Imagine a world where the very essence of life remained a mystery, where the fundamental units that make up all living organisms were unknown. The journey to understanding the cell, the basic unit of life, has been a long and winding one, spanning millennia and marked by the contributions of numerous brilliant minds. From the ancient Greeks who first pondered the nature of life, to the pioneers of microscopy who unveiled a hidden world teeming with tiny entities, each step has brought us closer to our current understanding of cells. This journey of discovery is a testament to human curiosity and our unyielding quest to comprehend the natural world. In this exploration of the cell's history, we delve into the remarkable discoveries and the brilliant minds that have illuminated our understanding of these fundamental units of life. Long before the invention of the microscope, ancient Greek philosophers like Aristotle and Thales of Miletus grappled with the concept of life. They observed the diversity of life on Earth and sought to understand the fundamental principles governing living organisms. While their tools and methods were limited, these early thinkers laid the groundwork for future scientific inquiry. Aristotle, for instance, proposed a theory of spontaneous generation, suggesting that life could arise from non-living matter. The ancient Greeks' emphasis on observation and logical reasoning laid the foundation for the scientific method, a systematic approach to inquiry that would revolutionize our understanding of the natural world. Their ideas, passed down through generations, would eventually serve as a springboard for the development of cell theory centuries later. The invention of the microscope in the 17th century marked a turning point in the study of life. In 1665, English scientist Robert Hooke published his groundbreaking work, which contained detailed illustrations of his observations using a compound microscope he had designed. Among his many observations, Hooke examined a thin slice of cork and discovered a network of tiny, empty compartments that he named cells, due to their resemblance to the cells of a honeycomb. Hooke's discovery of cells, although limited to the observation of dead plant cells, was a monumental step forward in our understanding of life's basic structure. His detailed drawings and descriptions sparked widespread curiosity and inspired other scientists to explore the microscopic world. It would take the work of subsequent scientists to unravel the true nature and importance of cells as the fundamental units of life. While Robert Hooke provided the first glimpse into the cellular world, it was Dutch scientist Antony van Leeuwenhoek who truly opened our eyes to the teeming diversity of microscopic life. A skilled lens grinder, Leeuwenhoek developed single-lens microscopes that were far more powerful than the compound microscopes of his time. With these instruments, he embarked on a lifelong exploration of the microscopic world, making astonishing discoveries that revolutionized our understanding of life. He was the first to observe single-celled organisms, which he called animalcules, in a variety of environments, including pond water, rainwater, and even his own saliva. His detailed descriptions and drawings revealed a hidden world teeming with life, challenging the prevailing notion that life could only exist at a macroscopic scale. His discoveries sparked both wonder and debate, challenging long-held beliefs about the nature of life and its origins. The 18th century saw a surge in scientific exploration, with advancements in microscopy and a growing interest in the natural world. Scientists continued to refine microscopes, leading to more detailed observations of cells and their structures. However, a comprehensive theory that unified these observations and explained the fundamental importance of cells was still lacking. During this period, scientists made significant strides in understanding the chemical basis of life. The discovery of elements like oxygen and the development of techniques for analyzing organic compounds paved the way for a more detailed understanding of cellular processes. Researchers began to recognize that cells were not simply inert chambers, but rather dynamic entities with complex internal structures. it wouldn't be until the 19th century that a series of groundbreaking discoveries would solidify the cell's place as the fundamental unit of life. In the 1830s, two German scientists, Matthias Schleiden and Theodor Schwann, independently made groundbreaking observations that would lay the foundation for cell theory. Schleiden, a botanist, studied plant tissues and concluded that all plants are composed of cells. Schwann, a zoologist, extended this observation to animals, concluding that all animals are also made up of cells. Their combined work led to the development of the first two tenets of cell theory. 1. All living organisms are composed of one or more cells. And 2. The cell is the basic unit of structure and organization in all living organisms. These principles revolutionized the way scientists viewed life, establishing the cell as the fundamental building block of all living things. While Schleiden and Schwann established the importance of cells as the fundamental units of life, the question of how new cells arose remained a subject of debate. Some scientists still clung to the idea of spontaneous generation, believing that cells could arise spontaneously from non-living matter. However, in 1855, German physician Rudolf Virchow proposed a radical idea that would forever change our understanding of cell division. Based on his own research and the work of other scientists, Virchow famously stated, "Omnis cellula e cellula," which translates to all cells from cells. This profound statement asserted that new cells could only arise from pre-existing cells, effectively refuting the long-held belief in spontaneous generation. Virchow's insights into cell division paved the way for further research into the mechanisms of cell replication, eventually leading to the discovery of mitosis and meiosis, the two fundamental processes by which cells divide and pass on their genetic information. While Virchow's work provided strong evidence against spontaneous generation, it was French scientist Louis Pasteur who delivered the final blow to this long-held belief. Pasteur, a pioneer in microbiology, conducted a series of elegant experiments that definitively demonstrated that life could not arise spontaneously from non-living matter. In his most famous experiment, Pasteur used swan-necked flasks filled with a nutrient broth. The flask's unique shape allowed air to enter but prevented dust and microbes from reaching the broth. Pasteur boiled the broth to sterilize it and then observed that no growth occurred in the flasks, even after extended periods. Pasteur's experiments not only disproved spontaneous generation, but also paved the way for the development of germ theory, the understanding that microorganisms are responsible for many diseases. The combined work of these pioneering scientists, from Hooke's initial observations to Pasteur's elegant experiments, has culminated in what we now know as modern cell theory. This fundamental principle in biology states that 1. All living organisms are composed of one or more cells. 2. The cell is the basic unit of structure and organization in all living organisms. And 3. All cells arise from pre-existing cells. Modern cell theory provides a unifying framework for understanding the diversity of life on Earth. It emphasizes that despite their vast differences in size, shape, and function, all living organisms share the same fundamental building block, the cell. This theory has revolutionized our understanding of life, providing a foundation for fields such as genetics, molecular biology, and biotechnology. As we continue to explore the intricacies of the cell, we gain a deeper appreciation for the beauty, complexity, and interconnectedness of all living things.